Taxi. What's all that?
He's coming.
Hello.
Hello. I feel like I should be saying good morning. I keep forgetting it's like 1 o'clock already.
It's noon for me, so I'm right on that fence of like, is it good morning, good afternoon? Yeah.
Hi, Alexandra.
Hello, how are you, Amber?
Good. I have Maxwell here. You can't really see him, but here's his arm.
How are you, Maxwell?
Do you want to tell her you want to be a mermaid?
No.
He wants to be a mermaid when he grows up.
Stop telling us.
Stop telling.
No.
What?
It's a
Is
secret.
it a
It's a
secret?
secret dream.
Yeah. Oh no, Dad. What's that?
That's a bird's cup. That's our neighbor's
A
cup.
dog?
No, it's not a dog. Dogs can't drive cars. So where are you going for this summer? You said you were going out of the country.
Yeah, I'm gonna be in the Netherlands for about, I guess, a little over a week for a course at one of the universities, Utrecht. So it's on translational medicine, so very relevant to what I do. So I'm gonna see how it goes, but I'm really excited. I've been in like Amsterdam airport, but never like actually visited the Netherlands. So I'm excited to also have like 3 days to kind of sightsee before I have to head back.
Oh, I am so jealous. That is so cool.
Me too. I'm jealous too.
Yeah, I love to travel. So like anytime there's an opportunity and someone's like, does anyone want to go? I'm like, sign me up. Like, please.
No, like. Mommy, what's that baby?
Sorry. Had to get the Little Mermaid. So a lot of parents are working during the day.
Okay. Yeah.
And so we plan to present this at a family meeting. And then also, one of a favorite parent is here. This is Andrew, and he's calling in from Canada, from the great land of Canada.
I like that. The great land of Canada. That's catchy. I hope that catches on.
Isn't
Good to
it?
see all
So
of you.
Andrew has an affected daughter, and he is also just incredibly smart, like computer level smart, and very involved. So this is fantastic. And you kind of met Becky. Becky runs most everything Hao-Fountain related. Becky, would you like to give an introduction?
Sure. My name is Becky and my daughter Ella is 12 and she has Hao-Fountain Syndrome. And like Amber said, like I do a lot of the social media, the website, communications with the parents, that kind of stuff. Just all the fun stuff, I guess.
Yeah. That's awesome. That's very much needed. So kudos to you to getting all of that up and running.
And most everybody knows me, but my name is Amber Freed, and I am a rare disease mom myself and helping other organizations. And I have been inspired by Matt Light and everything that is happening at the University of Alabama for as long as I can remember. For those of you that don't know him and will watch the recording a little later, Dr. Mike was a professor of computer science and pharmaceutical chemistry at the University of Utah. And he had a little boy with a variant. It was very hard to know what Bertrand had at the time. After a long diagnostic odyssey, Bertrand was diagnosed with a rare condition in GLY1, and Matt wrote a viral blog titled "Hunting Down My Son's Killer" that led him to discover other patients. Him and Bo Bigelow are very similar in the sense that they have an amazing writing ability, and they are just these fighter dads that came out to find more patients and make a difference. So I will now pass it along to Alexandra to fix anything I said incorrectly about MapMind and give a little background as to everything that is happening at University of Alabama, where I should add MapMind now runs this center.
Yeah, absolutely. And awesome introduction. You covered everything just great. So as I set up, let me share my screen. Oh, could whoever's the host, could you enable screen sharing, please?
Okay, I always forget how to do this part. This is the,
It might
I
be
go to
under
part-
security or something. It's usually, I think if I remember correctly, there's usually like a little security button at the bottom, and there might be a pop-up that has like allow others to share their screen or something that needs to be checked. Okay, it looks like it
all right,
works.
can you share? Okay.
Yes, okay, let me put this into presenter mode. Okay, so do you all see the full slide deck? Okay, great. So I know a lot of individuals will be, I guess, watching this recording. So good morning, good afternoon, whatever time point you are. I'm Alexandra Fukshinska, and I am the program manager at the UAB Hugh Call Precision Medicine Institute at the University of Alabama at Birmingham. And I'll be presenting on artificial intelligence-driven drug repurposing for rare disease. So just an overview of my presentation. I'll first introduce the role of artificial intelligence, or AI, in biomedical knowledge. Then I'll explain how my group's tool, Medicanron, works, and also get into a little bit of our process and provide an example of our process in action. So before getting into all of the technical stuff, I know many of you are aware finding treatments for rare disorders is challenging, and there are several reasons for it. I've kind of highlighted 4 in particular. One is limited understanding of disease mechanisms. So even if we know the gene or the mutation that's causing the disorder, we often lack just knowledge about how that gene functions in the body and how it interacts with other genes and proteins. Another reason is small cohort sizes, which is particularly problematic for rare disorders, because small patient populations make it difficult to, convince researchers and companies that there's a need for a treatment, and to also gather sufficient evidence for a treatment working that for, potential FDA approval. Then we have the little incentive to develop new treatments, and that goes along with the high costs of developing new drugs. So before this presentation, I was Googling to see what the current, I guess, estimated cost of developing a new drug is, and it's actually gone up, which is very concerning. But that cost still lies between like $1 to $2 billion, which, this high cost definitely discourages pharmaceutical companies from investing in new treatments. And then lastly, kind of inter-individual symptom variability. I feel like that's a tongue twister right there. Symptoms and severity, as you all probably can tell, even within the same, individuals that have the same mutation, you can see the wide variety between individuals, which complicates the process of identifying biomarkers and outcome measures, so something you can measure to determine whether or not a treatment is working. So I know I kind of started on a like Debbie Downer slide, but despite these challenges, I hope to kind of excite you with like how AI could help overcome some of these obstacles. So why AI in biomedical knowledge? First, we have the problem that there's so much information being published. I think it's like approximately 2 million new scientific papers published each year, and that's an insane amount of information. I have yet to meet anyone who reads every single paper, understands it, and has, memorizes all of the information of each paper. If you know of someone like that, please connect me with them. But I think it is impossible to keep up with, particularly our human brains. And that brings me also to my second point, because it's so hard to keep up with, sometimes knowledge gets forgotten or uninferred, which I know is not an actual word, but I use it to mean that there are certain connections and relationships between like pieces of information that can be drawn between like, say a group of papers. But unless, someone read every single paper, remembered every single detail from that paper, maybe that connection is not quite made. So yeah, it's definitely hard to one, remember every single piece of information, and two, be able to kind of infer some relationships. And then lastly, getting that knowledge, this information that's contained in, not only papers, but databases, data sets, into a searchable format that is also user friendly, is a major obstacle. So these 3 points are where AI helps us out. Before I get into AI, just a quick recap of drug repurposing. If I had to define it, I would say it's like giving a second life to medications that were originally created for one condition, but finding ways that they can, help with other disorders. So this helps us prevents us from starting from scratch to develop a new drug, which, as I said, would cost between $1 to $2 billion. And letting scientists explore existing medications to see if they can be effective for different disorders. And this approach also saves time and money, since these drugs have been tested for safety. So definitely if it's gotten FDA approved and it's used for something, that means we know about how it works in the human body, which takes usually the most time and effort in clinical trials. So what AI helps is making this process more efficient, so AI can quickly sift through millions of scientific papers, data, and research findings to identify potential new uses for existing drugs. And it can look for these hidden connections, the uninferred connections, as I said, and also patterns that, unless a human had read every single paper and knew every single detail, we would miss. So this AI and the AI tool I'll be explaining could kind of help suggest drugs that could be repurposed to treat rare or hard to treat diseases. And it also helps, researchers find promising drug candidates much faster than traditional methods. So how does it work? We start off with knowledge. So if I took our tool and like popped the hood and tried to show you how it kind of looks underneath, it's pretty much if you turned, I'm sure a lot of you have seen a scientific article and have maybe tried to like read through it. And it might have a lot of information, way too much information, lots of jargon, but all of the terms and connections that are made, if they were represented in just dots, it would, let's say it would look like this slide. And I have the different colors representing like different categories. So maybe one color is genes and another color is like drugs that are mentioned, and another color is maybe pathways or something. But like for this, like, for example, if I had the statement, chocolate is delicious, I would be connecting two dots, one representing the term chocolate, and then the other one representing like delicious, and I would have it connected there. But in order to like make sense of these different concepts and these different categories of concepts, we need to like provide some structure to it. And once you kind of have some structure, you can start, making those relationships a little bit more clear. So each line here that's forming is like a connection between one concept and another that was made, within a statement of a paper or something. And each piece of knowledge is unique. And if we zoomed in on like one of these, we could see that, a concept represented by the light green dot interacts with the concept represented by that gold or brown looking dot. So there's some sort of interaction there. Sometimes it can also get a little bit more specific. So it can, and also it could be directional. So the concept of the light green dot could inhibit the concept represented by that gold brown dot. And there's some sort of direction there. So this structured form of like these dots and the relationships between them is known as a knowledge graph. And there are different knowledge graphs for different knowledge sets. And we feed that into our tool. So now what does our tool do with these knowledge graphs? It can do a little bit more complex relationships. So if we start off with this dark green concept, and we see that it is connected to this concept represented by this yellow dot, that is in turn related to this light green concept. By inference, our human brains are like, okay, if green related to yellow, yellow related to light green, then dark green should, be related back to light green in a way. So by inference, first and third concepts are also related. And if we go even one more step out, looking at that gold brown concept, even though it's a little bit farther out from that dark green, there is still some sort of relationship there possibly. So this more complex thinking is what a knowledge reasoner does, and that's what our tool Medicanron does. And you might be thinking, what do dots and lines have to do with biomedical knowledge? If I isolate like that particular example, and remember each dot, represents a concept. And I told you that, let's say the dark green one was a disorder, the yellow one a pathway, light green a gene, and the gold or brown one is a drug. There might be papers that are linking that, okay, this disorder disrupts this particular pathway. This gene is associated with this pathway, and then there's this drug that activates this gene. And this could represent like 3 different papers that establish these relationships. But if we, reason or infer some of the other relationships, we could infer that maybe this gene, through its association with this pathway, may be related back to this disorder, or this drug, through its impact on this gene, could impact the pathway and maybe the disorder. So again, if a human were to like get to this point of making these kind of inference relationships, they would have had to like read those specific papers, remember those specific details, and be able to connect like those concepts and think about it kind of more on higher order thinking. But knowledge reasoners help us out by trying. And I say, I want to emphasize trying because they're not perfect by any means, but they try to help us make these kind of uninferred connections. And this is also different from, I guess, some of the more AI tools that are out there. I know I like play around with like the free version of ChatGPT, and it's not been very successful at this. Maybe the newer versions are. But even a Google search, if you think about Google, when you put in something and you hope to find like the most direct results in Google, but if, let's say, your direct results don't give you back what you're looking for, Google's not going to give you like the inferred result findings. So this is a little bit more complex and where the AI kind of helps us out. So our process and how it looks like, we start off almost always with a gene. And sometimes if we do know enough about a gene to know that maybe that's not the ideal target, maybe there's something like downstream of it or upstream of it that we might want to target, then we go after that. But usually we have at least a gene target or maybe a pathway or something. Then we like to consider the direction of regulation. So it's important to know, do you want to activate something or maybe inhibit it? Because sometimes there are some things that, just because our body's trying to compensate for a particular loss or gain of function, it might be doing it. So it might be actually benefiting us. So we don't want to disrupt that compensatory mechanism, but maybe there's something else that is dysregulated that is causing, certain symptoms, or conditions that, maybe we can target that. But knowing which
My
direction to go
question
into
here.
is important. Yeah.
Okay, I don't mean to, is it okay that I interrupted?
No, you're totally fine. Yeah. Please ask.
Okay, so this is like a perfect time for this question. In USP7 kids have too little of the protein, but if you have too much, then it's really associated with cancer.
Yeah.
Do you find that consistent with a lot of genetic variants,
Yes.
that there's like a Goldilocks effect?
Yes, there is certainly a Goldilocks effect for a lot of genes. The dosage, we like medications have doses. Genes also have a doses. So there's the sweet spot of like too little is not good, and you have typically, and it's like a rare disorder, but too much could lead to disruptions that might increase someone's risk at developing cancer. Of course, it all depends on, certain like other things outside of genetics, so lifestyle, diet, environment, and that sort of thing. But some genes do have that sweet spot of like, we want the expression to be increased, but also not too crazy increased levels, because that also could be related to another disorder. So we've also seen genes that there's loss of function and gain of function mechanisms to a disorder. So you definitely don't want to cause one or the other. You want to get to that sweet spot. So that is important. And that is also where we rely on researchers who are hopefully looking into, how much upregulation is needed to see maybe a therapeutic benefit, but also like how much is also too much.
Yeah, that's been such an interesting situation for Hao-Fountain, because there's so much research on treating cancer, having too much P53. So a lot of the new drugs that are coming out are talking about reducing the amount of the protein, and we're like, okay, can you do that backward?
Yeah. Yeah, it's a good point. And sometimes that's, where sometimes some collaborations happen. It's like, okay, exactly like you said, if you have found a way to inhibit it, is there also a way to potentially activate it? Yeah, very good point. And so that's why, we try to be very careful about looking at, do we want to regulate this and in which direction? Because we've also found that sometimes it's easier to inhibit things because drugs do a good job of tracking their target and inhibiting its function. It's always a little bit harder to maybe activate something, but there's it definitely varies from case to case. So yeah, that's a great question. I'm glad you brought that up. The other thing that we have to do is also we have to sort and review any hits that we get back. So sometimes like I provide a quick example of a issue I've run into is that like I was looking up the CMAS gene, and at some point, it was also known as the CSS gene. Someone had referred to it, or maybe it's another name known in that community. And when I was looking it up, we kept getting results, and I, would look at the papers associated with those results to kind of see if it's a true positive hit. But those papers were about cesarean sections. And I was like, okay, this has nothing to do with this gene. And then I realized that CSS is also an acronym for cesarean section. So the AI was trying to be very comprehensive and giving me back information that, it was like, oh, this gene's also, another name for it is CSS. So I'll pull in those CSS papers as well. And that makes us have to go through some of the results to make sure they're actually related back to what, the intended gene target was. So as for like the type of hits that we get, sometimes we get lucky. And if we start off with a drug target, sometimes we get a direct a gene target. Sometimes we get a drug that's directly associated or known to regulate that particular gene target. And we call that like the direct or one hop, because you just kind of hop once. But sometimes we can also look at more indirect approaches. So let's say there's, a gene or a pathway that's known to regulate our gene target of interest. Then we also get back results that show that drug or any drugs that, go after that pathway that then regulate the gene of our intended gene. And this is more of an indirect approach, and we call it kind of the two hop, because you kind of hop twice over before you get back to your gene. So once we have hits to look at, you might be wondering, like, are they ranked or prioritized in some way? And they are, depending on the tool that we use. Sometimes there's like a scoring method that brings in a lot of mathematical computations to kind of give back a score. But one of the ways it's scored is also just how many publications are pulled. And so with each drug, if this represented like 5, different drugs that came up, some might have only like 1 publication associated with it. Some might have 5 or 9. And so sometimes the natural way of like ranking some of these drugs is just based off of how many publications are associated with it. But of course, that also, requires us to review some of these hits manually just to make sure that, the one with 9 publications that tie the gene and the drug together is not a case where, it's talking about cesarean sections versus the actual gene that I'm looking for. So I wanted to provide like a example of Medicanron in action. And this is one case that we had worked on with another rare disease, the ADNP. And it's a known to be like I guess the mechanism behind it is that there's a deficiency of ADNP, so like a haploinsufficiency. So they were interested in increasing the expression of ADNP. And luckily, there was one drug that had come back with a direct relationship back to ADNP, and that it was reported in some paper published probably several years before we were looking at this for a rare disease foundation, was the drug ketamine. And so any results that we get, also, we kind of have our own medical director and clinical team that provide the clinical insights into like, is this, even something worth considering for drug repurposing? And after also several discussions with the researchers and clinicians working with the foundation, they put it to, they did some, a little bit of preclinical research, and then went on to start a clinical trial looking at treating with very low dose ketamine. And this was the first, I think, study of ADNP syndrome, and the preliminary clinical evidence show that there was some improvements in like social communication, ADHD symptoms, restricted repetitive behavior, speech, activities, and kind of daily living. So that was really encouraging. And as you see the stat article, it says it was like a nudge from AI. And although we wish, every single disorder we worked on had this kind of story behind it, sometimes our findings serve as kind of the foundation of, next steps. And one of those next steps could be like drug testing in animal and cell models. Because sometimes our hits, as encouraging as they can be on paper, we would prefer to have some sort of experimental validation of our screen, and therefore we like to work with researchers, particularly those that have cell or animal models with a good readout, to kind of test some of the drugs that come up in our hits. And see if any of them have a positive effect on the disease before ever getting to the point where it could be used for like an N of 1 trial. And so N of 1 trials, I think a lot of people are more familiar with that, but conducting on a single patient and used to kind of determine the optimal intervention, usually starting off with a very low dose, seeing if there's any benefit there, and then, going step by step, maybe increasing the dose up to a point where, okay, do we see any benefit? Yes or no. So all of our findings, we work and collaborate with research groups and pass them along to anyone who's interested in receiving any hits and maybe following up on them. And we also love to hear back if any did pan out and show any benefit in this cell or animal model, because that's one very exciting for the whole community. And if there is something that is easily accessible and has been, especially used in like pediatric patients before, then maybe seeing if there is a family willing to maybe try it out and see if they see any benefits. But yeah, I know I covered a lot of things in a short span of time, but if anyone has any questions or anything, I'm happy to answer or go back to any part of my presentation to clarify any points that I made or anything like that.
I have a question. So a lot of the families have been asked to donate blood or a skin punch. And one of the families was wondering, how is this useful in research?
Yeah, it is definitely it's kind of going back to like one of those first slides I showed with the what are the limitations to finding treatment options, is the understanding of a disorder and blood and cells, like the skin punch biopsies and getting fibroblasts and that developing into like iPSCs and other cell lines as well. Just give us more insights into the disease mechanism. And it's definitely helpful to have a wide range of different variants, and also, those who might have more severe symptoms versus those that have milder presentation, because all of that gives us more information about a particular just disease disorder and maybe the mechanisms, and shines a light on maybe some new drug targets that or gene targets that we can go after with our tools. So we rely heavily on the researchers who, are using blood samples and are using like skin punch biopsies and patient cell lines to maybe answer more questions and maybe get more insights into potential disease mechanisms. And then, they publish their results or, give us some insights that they've learned from those samples. And we take that and feed it into our tool. So the better information and better data we have, the more, targets we can go after and the more hypotheses and hits we can potentially generate that then could be tested.
Oh, that's really cool. Especially because it never hurts the kids.
Yeah. So yeah, it's like we want to be as inclusive as possible. And if there's something that, can work for as many patients, then, we would love to have as many people represented in the data that gets us to the point where we're making those, queries and running those things in our tools, so that we can, benefit the greatest number of individuals.
Yeah, absolutely. And I think that everybody's hope is receiving some type of a hit on something that can be easily repurposed or is easily accessible.
Yes, that is the ideal. Sometimes, ketamine is not something that anyone would be like, oh, okay, great, let me take my, child to the doctor and get some ketamine. It's usually used for like pain. And so this definitely required a lot more discussions, and a little bit of pre-testing to see if like, okay, does this seem to hold in, I think they might have tested patient cell lines or something. They did a little bit of testing before they ever, pulled the trigger on like, okay, let's try to make a more formal trial here. So sometimes we do get results that are, encouraging that it is something that, is accessible, could be prescribed. But it always helps, I know, the clinician to also have some sort of experimental validation, whether in a cell model or an animal model or something like that, to make it easier to make that decision to maybe try a low dose of something. So it's it's really exciting when we get to that point. But there's also a lot of the times we get some very interesting results back that clinicians are very hesitant to even start, and so that too we would want to see first tested in a preclinical model before ever thinking about trying out in a patient.
I have a quick question.
Yes.
I don't think you mentioned it in there. I saw that the ketamine, like on that particular case, the trial on people, on children, was for those 2 years. But how long before that trial did it take to get to that point? Like, just out of curiosity, like how long was that time frame?
Yeah, that is a great question, and I don't know off the top of my head. This was
Okay.
before I joined the team, so I am not as familiar with kind of the preclinical trial. I think I joined as that clinical trial was just getting off the ground. So unfortunately, I'm not able to answer your question, but I can definitely look into it and then let you know.
Yeah, I was just curious, like how
Yeah.
long it takes to get to that point of being able to, actually go ahead to people and try things.
Yeah, and I think it's highly dependent on, what is known about the disorder. If they do have a good, model already established that they're like, oh, okay, ketamine, let's try it on the cell line. Do we see any benefit and that sort of thing?
And I'm sure it also is dependent on what the actual drug might be too, because some might be more
yes.
harmful than helpful.
Yes, and dosing, same thing. It's like, okay, if we put, a very minimal amount of the drug, do we see any benefit? Because then some drugs are also very toxic at a certain amount. But sometimes you have to get to a certain dose of a drug to see a benefit in an animal or a cell line. But that if you convert that dose to what you would need for the human, it's absolutely like crazy amount. So you're like, okay, I can't like give this huge amount of drug to my patient. So yeah, there's a lot of variables at play. But that is a very great question. And I know there's some publications that have come out of this ketamine finding. So maybe even tracking down the timeline of those publications will provide some insights into kind of how long did it take to get to the trial stage.
I can speak from experience. It's also just really reliant on the type of drug. Oh, sorry, I lost video. The type of drug and how available it is. For other disorders, we have found a drug that's working really well, but it's incredibly expensive. And so even though like your primary care can prescribe it, well, it's hard to get it actually covered.
Yes.
But then I know some disorders that had a big hit on drugs as common as Paxil And their doctor is like, yeah, why not? It's already in the drinking water. Or maybe something that's available over the counter.
Mm-hmm. Yes, yes. That is a big component of, you know, just thinking about anything, even getting to a trial is already like a huge deal. But knowing where you want to start dosage wise and knowing if that is also an available formulation, because sometimes there are some drugs that are available, you know, only as a certain form, but it's like, okay, could we possibly compound it and like get it into another formulation that would be specific for, you know, if it's a dermatological condition, let's not like have them taking something that might affect their entire body. Let's just have it concentrated just to kind of treat, you know, their dermatological symptoms. So there's so much at play here with drug development, or even just using drugs in a clinical trial for a rare disease. Yeah, there's it's fascinating, but also there's just a lot of it creates a lot of obstacles to get to that point where you have something that you can test.
It's happening so much faster though now.
Yeah, that's the exciting part is drug repurposing is definitely making its mark in rare disease, but also in other disorders as well.
Becky, did you have any other questions?
That was the big one that I thought of during the presentation, but it was very, very good, very informative.
So I'm glad. I know it is a lot of like when people think AI and drug repurposing, they're like, oh, so you just throw it into Chat GPT. And I'm like, let's not do that because sometimes, you know, Chat GPT still has its limitations and sometimes hallucinations that could be very harmful. So our tools, I mean, have been in development for probably more than a decade or so. And they're constantly improving, and there's a lot of things that, you know, even the current new AI tools that are coming out could also help us out, kind of make our process a little bit more efficient. Like maybe the manual review process, maybe if we used AI to kind of help us out there and make that a little faster. So it's definitely exciting, the AI standpoint, but also just drug repurposing and all of the new initiatives and grants and money being applied to, you know, can we take medications that already exist and see what other disorders they could potentially treat?
Well, how nice would that be?
I
Thank
know.
you so much for your time today. We really appreciate it.
Yeah,
Thank you so much.
I appreciate the invite, and I hope everyone learned a little something. But definitely, if anyone has any questions, feel free to reach out.
Sounds great. Bye.
Awesome. Take care.
you.
Bye.